I hope you're enjoying this video series on enzymes and metabolism. What I'd like to do in this one is take the topic a little bit further and deal with metabolic pathways. We've mentioned all sorts of enzymatic reactions so far, but we haven't said anything about what really goes on in cells in terms of pathways. So what we've done, if we think about it, we've taken things like glucose and glucose, and we know it's a reversible reaction. The enzyme is maltase, and we make maltose, that sort of thing. That's an isolated reaction. And where that may happen in cells, that's not the only thing that happens in cells. What we typically get is a situation like this, where we have a substrate, let's just call it an A, give it an A for want of a better name, gets converted into something, and we'll call it B, which gets converted into something else, C, D, and so on. This is a pathway. In these metabolic pathways, typically an initial substrate is converted by an enzyme, we'll just call it enzyme 1, into another substrate, which gets converted by another enzyme into a third substrate. I'll just call it an S3 now instead of writing out substrate, and a fourth substrate after the activity of a third enzyme, and so on like that. That's more what happens, where the product of one step, B, is the product in the first enzymatic reaction. The product of one step becomes the substrate for the next step. And that product, because this is a product of this step, becomes the substrate for the next step, and so on like that. And we have an enzyme 4 here, and it gets converted into, I hesitated here, I was going to put D and then E, but I'm going to put F here because we've already used E to represent the enzymes. This is more what goes on in organisms. This could be the final product, maybe then. We'll just give it a P for a final product. I want to take a really well-known example and present that to you as an illustration of this. What we know is this. We know that cellular respiration is basically the reverse reaction to photosynthesis. Not that you can take one and just turn it backwards and becomes the other one, but if you look at the net reactions of these things, the net reactions is the reverse of it. So if we take, say, we'll put our glucose down here, C6H12O6 and 6O2, and we'll put 6CO2 and 6H2O. If we could take this reaction, that represents photosynthesis, where we take carbon dioxide and water, we can make sugar and oxygen out of it. We can't do it. Plants can do it. Cellular respiration is basically considered to be the reverse of that. The example I want to use is this one here, taking this and making that. Taking the sugar and the oxygen and trying to get carbon dioxide and water out of it. But before I do that, I want to come back to a point that this is cellular respiration is actually 3 different kinds of processes. Inside this umbrella, if you like, of cellular respiration, we have carbohydrate metabolism, which is where we actually take sugar here and metabolize it into smaller products. We also have fatty acid metabolism. Oops. Where we take fatty acids, and if you think back to what I said in an earlier video, I said we can metabolize fats and get energy out of them, or we could also metabolize protein. Our body doesn't like metabolizing protein. It's hard on the system. It's also a sign of of declining health. If you run out of sugar, you can metabolize fatty acids and you can use up some of your fat tissues that are in the body. If you were in a situation where you ran out of both of those, your body would revert to metabolizing protein. So if you think about people who are on, who are starving, you've seen pictures of people who are starving, they are nothing but skin and bone. Well, if you think about it, the bone's there, the skin's there, the subcutaneous fat is gone, and they're using up their muscle tissue. So that's the last in that series. This is the one we're going to look at right here, carbohydrate metabolism, not fatty acid metabolism, not protein metabolism. So what we'll do is, in a minute I'll start this, we'll take a look at sugar and the conversion of sugar into carbon dioxide and water with the presence of oxygen. The whole reason we do that is because we want to get energy. We burn carbohydrates for energy, and of course the sunlight has to provide that energy in to get that thing to go like that. So this thing, although we represent it like this, glucose and carbon dioxide makes that's like one-step reaction. There's one arrow involved in that. That's a huge, huge oversimplification, because there's absolutely no way you can take these two ingredients and mix them up in whatever condition you've got, and you're going to wind up with that. There are many, many, many steps to get to these products. Many steps. So this is a long metabolic pathway, and that's what I'd like to do. So I can take this off. So what we're really going to look at is carbohydrate metabolism. And I'm not going to get into the whole topic, into all the minute details of what goes on all the way through these pathways, because that would take several videos, and we're not going to go that far with it. But this is what we tend to think of as cellular respiration. Because that's the primary way that we get our energy out of our food sources. Carbohydrate metabolism. Really, it occurs in many cells. I can't say all cells, that'd be an oversimplification, but we're going to say it, I think we could probably say it occurs in most cells. Prokaryotic and eukaryotic. Remember, eukaryotic cells are those cells that don't have internal membranes. These cells don't have nuclei, they don't have mitochondria, they don't have all the organelles that prokaryotic cells have to make, for their own existence. They have ribosomes, but that's not a membranous organelle. So the eukaryotic cells I think I said that backwards. Prokaryotic cells don't have the membranous organelles. Eukaryotic cells do have the membranous organelles. And the reason that's important to recognize is that this process can occur in both of these. Although we know that the mitochondria, which are in these ones, are the powerhouses of the cells. By this expression, powerhouses of cells, they actually make most of the ATP. That's most of the energy, because the energy is in the form of ATP. But both of these kinds of cells can do it. These are way more efficient at it because they have mitochondria. These ones don't have the mitochondria. So what we have here is that it occurs in 3 metabolic pathways. Three of them. The first one is called glycolysis. Glycolysis occurs in both of those. Glycolysis results in a net production of 2 ATP. 2 molecules of ATP is all that can be produced from 1 molecule of glucose. So both prokaryotic cells, like bacteria for example, that's all they need. We are made up of eukaryotic cells. We would never survive if we could only get 2 ATP molecules out of each sugar molecule. The other 2 pathways are Krebs Cycle and the electron transport chain. These 2 only occur in eukaryotic cells. Because of the mitochondria. And these 2 combined, when you take the effects of both of them, result in the production of 34 more ATP per glucose. So now if you look at it, these things, the mitochondria are considered the powerhouses of cells because they get a lot more ATP production with mitochondria than just the simple 2 that you have with glycolysis. But what I want to do is I want to take a closer look at these reactions because our topic is metabolic pathways. How do you get this thing to become that thing, to become another thing, to become another thing? And that's what goes on in here. So looking at glycolysis. Glycolysis has quite a few steps in it. It starts with sugar, C6H12O6. The first thing that happens is it has to be phosphorylated. Gets phosphorylated, basically ATP is added to it. Two ATP get added to it. So what you get is this sugar, phosphorylation gets 2 ATP added to it. So what that does is basically it energizes the molecule. Now, wait a minute, we're supposed to be getting ATP out as a product. We just put 2 ATP in. You have to remember that because what I had, I just erase it now, is we get a net production of 2 ATP out of glycolysis. We're going to get ATP produced here at the end, but we had to put 2 in to get 4 out. So there's only a net production of 2. So this step here, this phosphorylation, is basically energizing the sugar molecule so that enzymes can work on it to do a number of things. The second thing that happens to this, and it doesn't, I'm oversimplifying it a little bit, again, because there are several steps in each one of these parts, but the second thing that happens to it is that it gets cleaved in half. It's called sugar cleavage. And if you think about it, this is a C6H12O6. We're going to take a C3 half, I'm not going to give you all the bits and pieces here, and ATP. And we're going to get a C3 part with ATP here. Oop, ATP. The third thing that happens is these 2 pieces, which are basically when they're produced, they're isomers of each other, but then there's an enzyme that takes one of them and converts it into another one. So it's a 1,3-glyceraldehyde molecule. It's its real name, but we don't have to know that right now. We won't get into that degree of detail. These things here, identical kind of molecules, they get converted into what is abbreviated as PYR, which, if we take the full name, it's pyruvic acid, or we call it pyruvate, which is the ionic version of it, because it's in a solution, it's in a cell, it's going to be ionic, but it is pyruvic acid. So these two things will make PYR. Notice that pyruvic acid is a 3-carbon molecule. I'm just going to put the C3 underneath it here. And then these things, pyruvic acid, if we're talking prokaryotic cells which don't have mitochondria, these things become products that get used in the cell in another way. If we're talking about eukaryotic cells which have mitochondria, pyruvic acid actually is converted into another molecule, which we'll get to in just a second here, and that other molecule goes into the mitochondria and gets used in Krebs cycle, and then in the electron transport chain. But this is what glycolysis is. So like I said, I want to illustrate this as an example of a metabolic pathway. And the real thing is more like about 10 steps, but I've simplified it down into these 3 components like that. I want to go back and look more closely at pyruvic acid too, because we have, I'll leave that list there. We have had this in another video previously. Pyruvic acid is supposed to, if oxygen is available, be converted into an acetate ion. That's what's supposed to happen here, given that oxygen is available. This is a 3-carbon molecule. Oops. Acetate ions are only 2-carbon molecules, or ions. What happens to do that is carbon dioxide is generated. And you know that in your cellular respiration, you're generating carbon dioxide. That was one of the products, carbon dioxide and water are given off, that sort of thing. So that still balances the number of carbons. What I said is that if oxygen is available, that's what happens. If oxygen is not available, we don't get the acetate ion forming. What we get instead is lactic acid forming. And lactic acid is a 3-carbon molecule. Lactic acid is that substance that builds up in muscle tissues if you've overexerted yourself for long periods of time, and basically your available oxygen is low. So you don't have enough oxygen to completely metabolize pyruvic acid into the acetate ions. It's kind of like if you have ever watched a marathon runner and the expression is they hit the wall. They've run out of sugar, they've run out of oxygen, they just don't function. They build up lactic acid in their systems. This, when oxygen becomes available, this can be converted back into pyruvic acid. This going, I should draw that a little bit differently. Oxygen allows it to go that way. No oxygen goes that way. The enzyme in here is LAD, lactic acid dehydrogenase. It's the substrate named after that substrate to there. That's what goes on in animal tissues. In plant tissues, because plants do this as well, pyruvic acid, plants don't get sore and stiff. That's not what happens. In plant tissues, if oxygen is available, that's all fine. Acetate. This is plants. oxygen's available, that's going to be just fine there. If oxygen's not available, see the difference? If oxygen's not available, you get alcohol. This is alcoholic fermentation. This is how we make alcohol out of plant materials. This is the process that goes on here. Alcohol is a 2-carbon molecule, ethanol, ethyl alcohol. If we had this situation here where oxygen gets added back into it, we can get back to pyruvic acid. If you take this process here, an alcoholic fermentation, and you add the oxygen to it, if you've ever noticed people who are making wine, they'll have a bubbler on top of it that allows the carbon dioxide gas to escape. They don't want the oxygen to get in, but if you were to add oxygen to it during this process, you're going to wind up with vinegar. It's a 2-carbon molecule. There's no way you can't get this thing to go backwards. You can't get it to go backwards because the carbon dioxide gas has already been evolved. It's being given off. This doesn't go backwards. This won't go backwards. So there you have a consideration of glycolysis and how you get it phosphorylated and eventually get to the pyruvic acid formation. Plant cells, animal cells, both do that kind of process. Here's the difference between fermentation in animal tissues, it's reversible, lactic acid production. Fermentation in plant tissues, not reversible, vinegar production. But we haven't gone into the rest of it yet. I'm going to take this part off, and we're just going to consider this. We'll just consider what goes on in plant or animal tissues here. And we have our acetate ions. What we have now is what we suggested is Krebs cycle going on inside mitochondria. So now, if this is a mitochondria, remember structure of mitochondria, they are organelles that have a double membrane, and they have a matrix inside, so you've got an outer membrane and an inner membrane, and then they've got a matrix. Along this membrane here, this inner membrane, are all sorts of enzyme complexes. And these enzyme complexes are exactly the location of Krebs cycle. Hans Krebs, with an S on the end of his name, got the Nobel Prize in 1953 for figuring all this sort of stuff out. What happens in Krebs cycle is quite simply that this acetate ion, which gets introduced into here, gets bonded onto, and I'll just take that over here, and I won't use the mitochondria. Here's the C2 part getting added onto. It gets added onto a C4 molecule. This molecule, it's oxaloacetic acid, although we don't have to get into the detail of it, because you get the C2 and the C4, it becomes a C6 molecule, which eventually gets degraded, C5. There's actually a couple of C4s in here, back to C4 molecule. And you can see what happens in here. Carbon dioxide is given off here because there's a loss of a carbon. Carbon dioxide is given off here, there's a loss of a carbon. And you wind up with the 2-carbon molecule you put in gets used up in the production of these 2 carbon dioxide molecules. But the essential part of this, and how we get all this ATP out of it, is in this changing of the molecules in here, we get a product. We actually do get a little bit of ATP out of one of these reaction steps in here. So I'll just put ATP gets produced directly, but we also get, it produces NADH2. Because if you think about it, the acetate ion is mostly carbon and hydrogen. It's got some oxygen on it. If we expel carbon dioxide, we still have to have some place for the hydrogen to go. And what happens here is NAD, and I introduced NAD in a previous video, that's that nicotinamide adenine dinucleotide. It actually picks up 2 hydrogen and it makes this, NADH2. NADH2 is known as a hydrogen carrier. And the hydrogen carrier actually transports this hydrogen into the electron transport chain, and it uses the electron transport chain, which is another series of enzymes in here, to take the hydrogen and remove the extra energy from it, because the energy isn't in here. The energy in the molecule is gone with the hydrogen. And each one of these is capable, there's enough energy in each one of these to make 3 ATP molecules. So if you take all of this together and you're trying to balance all the energy in it, you would have a total of 30, well, from here on, well, actually from here on, 34 ATP being produced, but we had 2 more being produced up there and we used 2, so we get a total of 36 ATP produced throughout. The details in here, like I say, are beyond the scope of what we need to deal with. It's important that you recognize that metabolic pathways is the way things are done. It's not a single step reaction where in a metabolic pathway you get one molecule becoming another molecule becoming another molecule and that sort of thing. And it's through the regulation of these pathways that cells do make their energy. In the next video we're going to take a look at how these pathways get regulated. I mean, we have a bunch of pathways in our body, but how do you know if this pathway is working, and how do you slow it down, or how do you speed it up, and that sort of thing? But that's coming up in the next video. Thanks very much.